welcome again. So today I want to tell you a little bit about another lesson that has to do with food chemistry and microbiology. It's to an extent a continuation of what we talked about at the last PD, but this one is more focused on the microbiology of bread spoilage or food spoilage in general. I showed the slides last week, so I'll probably be brief over here, but essentially food science is a good handle or a good latching point to convey a lot of science concepts. And it's also fairly easy to contextualize science knowledge related to foods, given that, well, food is in our daily lives, and we see the importance of food safety and nutrition in a lot of topics of common concern, be it our current pandemic, the environment, and also the social justice aspect of food as well. So today's class is about food spoilage. We'll be using bread as the model here. And so there are two things I want to go through. One is the use of experiment design and how that is useful in the study of spoilage, and secondly, the actual microbiology part of spoilage. So a little bit of disclaimer here. Well, what we're doing in the lesson is using sliced breads. We can use the bread from another lesson or just buy sliced breads, ideally white bread from the grocery. And so basically treat the bread slices to different moisture contents and then swap them with a certain surface, be it your personal cell phone, kitchen sink, towels in the shower, dirts outside, or your laptop computer. So those are surfaces that we expect to collect microbiome from. And then we'll put these bread slices into Ziploc bags. I highly recommend using Ziploc bags with sliders, because once molds start to grow, it grows well in a sealed environment. So yeah, pretty much using bread as a petri dish, as Jay mentioned. So we want to make sure that it's in a sealed environment, because we want to keep the moisture inside. And also, in the case where molds start to grow, most spores can be generated by the colony, and if we let them escape the bags, then that's potentially a health hazard, particularly for those who are allergic to mold. So in practice, those with sliders offers a better seal compared to the other type of Ziploc bags where you need to pinch it, because sometimes if you're not pinching it correctly, you may get a leakage. So the other important disclaimer to have, and this is important if you do use the lesson in your class or in your club, is students should not open the bags that has moldy bread in them, because of, again, the allergy issue, and also certain molds can generate mycotoxins, which are harmful to human body. They can be carcinogenic well, when it comes to chronic exposure, but even not, it can cause other illnesses as well. So yeah, we're not supposed to expose the moldy breads in the air. They should stay in the Ziploc bags, and when disposing them, they should go into whatever stream that goes into a landfill. So they should not go into a recyclable stream or a compost stream. The reason being, if someone ever asks, well, Ziploc bags, although they are made of plastics, they're not recyclable. And well, particularly when there's moldy bread in there, it makes it even less recyclable. And so the plastics are not compostable either. So basically they should go into a landfill stream. Okay, so this is just a basic design of the lesson. It's pretty straightforward. We are looking at the impact of 3 different factors on the growth of molds on bread surface. So temperature, moisture, and which location we're swabbing it on. I'm using location 1, 2, and 3 just to be general, but in your class, you can identify your own services, like for example kitchen sink or doorknobs, for example. So the moisture content was controlled by, well, either just without adding any water to the bread, or just sprinkling some water onto the bread surface. Usually, basically 1 tablespoon is probably good enough per slice of bread. And for a soak sample, you just soak that slice of bread in a container that has water in it, and make sure that's submerged. Wait for a couple of seconds and then take it out. That's pretty straightforward. Temperature-wise, warm room temperature and cool. So these are just really rough estimates of levels they can go with. So they don't necessarily have to be controlled super accurately. So the cool sample can go into a refrigerator. A warm sample, there are a couple of ways to do this, but you could put it in the back of a fridge where there's radiator. So that's probably one easy way to do it. Or you can attach it to maybe a vent of an HVAC unit. And so, yeah, you don't really need an thermal oven or incubator for this, but if you do have access to those things, then that'll work even better. I don't recommend going too high of a temperature because, number one, there may be too much vapor generation inside the Ziploc bag if temperature is too high, and that can compromise the seal, which will be bad. And also, too high of a temperature beyond 70 degrees Celsius can actually negatively impact the growth of mold because it's too warm. So basically, we treat the slices of bread, put them in bags, and then keep them for 1 week. Usually in 3 to 5 days, you can see some notable differences. 1 week is probably safe enough. And then dispose them. So I did not include this in the lesson plan, but it occurred to me that I should add a disclaimer here. So this lesson should be done with just regular bread, ideally white bread. So it's not recommended to be done with any flavored breads or sourdough, because those breads don't really support mold growth as good as white bread would. So we have essentially 3 factors, right? Temperature, moisture, and the surface we're swabbing it on. And each of these factors have different variables. We can call them levels, right? So we have different levels of temperature, we can have different levels of moisture, and different swabbing surfaces. So if we actually make samples of all possible combinations of these factors, we'll have 3 times 3 times 3, which equals to 27 different combinations. And that's a design of the experiment where we have all possible combinations. We usually call it a full factorial design. This is showing you a full factorial design with 2 factors only. So I'm just calling them factor A and factor B. And for each factor, we have 3 levels. So for example, if factor A is temperature, we have cool, room temperature, and warm. Yeah, so For the lesson itself, it's recommended that each student only look at 2 factors. So that's going to be 9 slices of bread, because 27 slices of bread is a little bit too much. But 9 is probably a good number for observations. So, okay. Now, this design is called a full factorial design, but in reality, particularly in the food industry, when we try to answer questions like, well, how do the environment affect shelf life? And the environment probably consists of many different factors. And if we try every individual combination of these factors, there just are too many experiments to run. So another concept in factorial design is to have a partial factorial. So what you're seeing here is a cube. I imagine that this is actually a cube-shaped lattice, right? You have 3 dimensions corresponding to each of the 3 factors, and you have 3 levels. But you only do those experiments that correspond to these dots. So as you can see from the picture, there are only 9 dots in there. Well, actually, no, those are not 9 dots. My bad. Those are 13 dots in here. Yeah, 13, 1, 2, 3, 4, 5, 6, 7, sorry, 15, yeah. So 15 experiments in this partial factorial, which means we're only selecting 15 conditions or 15 combinations out of the 27 total possible combinations. And so this is a reduced design, but usually such a design is enough for us to identify the impacts of those factors. So this is called a partial factorial, and we're carefully picking those 15 combinations of factors so that they're representative, or they can give us enough data to draw the conclusion we actually need. So I'll probably just stop there. We'll just introduce these concepts without actually diving further into how to analyze data if we're using a partial factorial design, because it's a little bit too complex. But yeah, these concepts are further elaborated in statistics. So students will learn about these techniques as they move on and take classes in those disciplines. So, so what factors do affect mold growth? Well, moisture does play a factor. Really, the higher the moisture, the more favorable it is. Temperature is also important. So low temperature slows down mold growth. And the availability of a carbon source also matters. So usually sugars and starch are the preferred food of choice for mold. The presence of nitrogen source can also help the growth of mold, because nitrogen is involved in the synthesis of nucleic acids, amino acids, etc. So that's also an important nutrient for mold growth as well. The absence of sunlight is important, particularly during the early phase of mold growth, because UV lights can suppress the growth of mold. But once the mold has already formed a colony, then sunlight doesn't really make a very big difference. Okay. So once the mold has already started growing, you'll probably see things like these. These are just some sample pictures. So usually the sample that has no water addition and that has never touched any surface will have the least mold growth, as you can see on the picture in the middle. So the actual color of the mold can vary quite a bit depending on the actual organism that grows on it. So usually you'll see some dark green, black, or white colonies, but sometimes you see orange, pink, or in this case yellow or brown colonies there, which indicates aspergillus growth. So yeah, it really varies, and honestly, it's not really possible to identify the microbe just by eyeballing the appearance of the colony. Usually, some microbiology techniques is needed to actually identify the organism. So there's a question in the chat: is mold using aerobic or anaerobic processes? That's a good question. They can do both. So they have the capacity to generate energy both via aerobic or anaerobic pathways, although they grow particularly well under aerobic conditions. So the diplobac themselves, they're not exactly airtight, so air can still permeate across the bag, and there's residual air inside the bag as well. So yeah. That's pretty much what the lesson is about. Students will observe after a week the mold growth, and they can compare the growth of colonies on those slices of bread semi-quantitatively or just qualitatively, looking at how large the colony has become and the color of those, and see whether the different factors that they use for their experiments actually made any difference. And then there can be a discussion about, well, what does the experiment results say about, what kind of environment favors mold growth, and what does that tell us about maintaining a longer shelf life for foods, right? Just a little bit more on fungal microbiology. So there are many types of molds that can grow on the surface of breads. I'm just listing 3 here because they're the most common ones. The first one is called Rhizopus solonifera, and it is also called black bread mold because they form black colonies like these and here. So they usually form these round black dots. You see these dots on grapes and some peppers as well. They're usually the same organism. So if you look closer, or if you put the sample under a microscope, you can actually see the microscopic structure. So the white strands are the mycelium, which is coming out of a germinated spore, and then on the top you have these little black circles that are basically little spheres that where spores are being produced. So penicillin is another commonly found bread mold. It's usually gray or white in color, sometimes blue. And as you have suspected, penicillin is linked to penicillin. So penicillin is an antibacterial agent produced by penicillin mold that suppress bacterial growth, which gives the fungus itself an advantage or exclusive claim of the carbon source. So penicillin also, or certain species of penicillin also confer a unique flavor to the food. So in some cases, they are purposefully cultured in food. One example is blue cheese. So you probably noticed some mold growth on blue cheese and the unique smell. Yeah, exactly. So those are actually from penicillin growth. Although penicillin, certain species of it can produce mycotoxins, and they can have adverse health effects on humans, and they can be carcinogenic too. So that's another risk factor. But generally speaking, Rhizopus and penicillium colonies, they don't they don't really alter the sensory properties of the food that much. So if you do eat them, it's probably not a big deal. You won't get food poisoning or anything. So cladosporium, this is a unique one that I will highlight because it's commonly found in bread and delivers a very strong moldy smell. So the moldy smell that you sense from moldy grains and also moldy breads is most likely responsible coming from cladosporium. It also releases allergens as well to a various amount compared to penicillin or rhizopus. So yeah, this is the one that's mostly most responsible for the allergenic effects of mold colonies. It's usually dark green in color. Sometimes it can be black too. So they can also look fairly fuzzy like penicillin, so that this one could be cladosporium because it looks fuzzy, and in comparison to penicillin, they form spot-shaped colonies, whereas cladosporium can be quite irregular in shape. Anyway, so these are just some notable examples. There are a lot more types of organisms that can grow on breads in the number of thousands. So these three are probably the most common ones. A question about how large the slice of bread has to be. It doesn't really matter as long as the bread can still maintain integrity after mold growth. So what we don't want to happen is after mold growth, the bread basically fall into pieces or turns into scrambles. So I would recommend a minimum size of 1 inch by 1 inch. It's still large enough size that I can see progression of mold growth. And yeah, so although it's recommended that we only, quarter pieces, exactly. It's recommended that we keep one piece of bread sample in each Ziploc bag, so that they don't interfere each other when they're growing. So that's another consideration as well. We're probably using a fair amount of Ziploc bags in this activity. Another interesting fact about bread. So when we see a loaf of bread, for example, already infected with molds, this is just the surface. So mold can go far and deep inside the bread itself, particularly the mycelium part, and the surface is just a very small part of the colony itself. Okay, so just want to show you some interesting data. This is showing the presence of fungal species inside a kitchen. So basically, a few different families of fungi are being tested here. I believe this is via 16S. So the samples are collected from swabs of 4 different locations, and you can see that they share some common features, although the drain is probably a little bit different when it comes to frequent taxa. And yeah, so you can see that the Agaricomycetes, the one at the very top, that's the one that's linked to mushrooms you see in the yard. And so if you go down a little further, you can probably see. So Uroshiomycetes, that's another yeast. Saccharomycetes, that's the yeast that's used for bread making and brewing. But yeah, so basically, the microbiome on, well, kitchen surfaces, for example, can be quite complex. And when it comes to growing on breads, certain types of molds or fungus species that are more advantageous. That's why we see more of them rather than others. I think I have another slide here. Well, yeah, I think I removed the other slide because it's a little bit hard to interpret. But when we're actually using the lesson, the molds can actually be quite diverse, and the shell colony can be quite diverse as well. So that's another way how we can confer the idea that, well, in our living environment, there are many different types of microbes present. You can see them on our bread petri dishes. So, okay. A couple of questions that you may expect from the class. Where does the mold come from? Mold spores are very common in our living environments, particularly in areas where there's high moisture. So Oregon is now, particularly in winter months, to have a high level of mold spore counts in the air. There are databases and sources where you can see. It's similar to weather forecasting. They can see forecasting of mold spore concentration in the air, depending on region. So these mold spores, once they land on the surface, they can start to germinate and grow into a mold colony. So usually they start to grow a strand-shaped mycelium, and that extends across the substrate and assimilate nutrients, and then they make more spores. That's how mold grows. Now, the germination process requires moisture, which means mold cannot grow in a moisture-free environment. So it does need that. But once the colony is already established, it has some resistance against low moisture because it forms a biofilm that slows down water loss from the colony. Okay, what happens if I consume moldy bread? You probably wouldn't get sick necessarily, although mold releases these odor compounds that probably will make you nauseous or even vomit if you eat heavily infested bread. But you probably won't have anything more severe than that. There are a few examples here. There are certain molds that generate lethal mycotoxins, so that should be made aware of. So not all molds are mild enough to cause a nauseation response as the only response. It can be lethal. Although moldy foods are actually common, so blue cheese is an example. So there's another runner of cheese called brie cheese, B-R-I-E, brie, that also has mold growth on its surface as well. So yeah, the use of mold is very common in the food industry to introduce additional flavor. Particularly, it's a common practice in ethnic foods. The use of molds is also common in the coffee industry, where coffee beans are fermented as a part of the production process for coffee. So yeah, mold is not actually all bad, but yeah, definitely do not eat the bread after you're done with experiments. And well, for the sake of food safety, moldy foods are not for consumption in general unless the mold was cultured in a controlled environment and it's well established as a processing method. For example, blue cheese. How to suppress mold growth? This is an important question when it comes to shelf life and food safety. So there are a couple of approaches. One is the addition of antifungal additives. They can be synthetic chemicals, for example, sodium benzoate. or acids. So usually lactic acids and citric acids are food-based antifungal acids that are used in foods. Certain flavor compounds and the fruit extracts can have similar effects. So examples include citrus flavor, ginger flavor, and garlic flavor. So gingerbreads and garlic breads are usually more resilient or resistant to mold growth because of those natural antifungal components. Another approach is to use biological methods. Essentially, lactic acid bacteria, which produce lactic acid and lower the pH of the food. Mold grow the best between pH 5 and 7, so the lower the pH, the less favorable the growth condition is. This is also why it's hard to grow mold on sourdough. They tend to last longer. So to control molds, you can either add these acids as an additive or just use a sourdough starter. So sourdough starter is actually a combination of lactic acid bacteria and yeast. So at the ends, both organisms will grow on the breads, and that confers natural resistance against mold growth. Another question from a chat: coffee beans, are they using molds or just the biological process? I'm not 100% certain, but I think it has mold involved. And yeah, so I do need to look into this a little bit more. The fermentation of coffee beans in general, it's all good. The fermentation of coffee beans in general involves molds. So that's a process where the actual mold growth actually alters the flavor of the coffee beans, and yeah, so there are certain ethnic foods that involves culturing of mold as well. One example is fermented tofu. So basically, tofu can be cultured. Yeah, so basically, tofu can be fermented with mold as well, just like how cheese could. So that's an example of food culture with mold. That's actually all I have for the lesson. So yeah, maybe we can open this up to more questions.
That sounds great. Well, thanks for that lesson, Glenn. I have a couple questions. Thinking about the experimental design and setup as being part of the lesson for students. That's part of the next generation science standards, as I think we've discussed before, is developing or planning and carrying out investigations. And I think this is a perfect example of how students think about that and set it up. And you talked a little bit about it at the beginning. As students are thinking about some simpler aspects of it, how many replicates should they be doing? What sort of like controls should they be setting up? I like the idea of using smaller amounts of bread, especially if they're going to be using the bread that they potentially baked from the first lessons.
Right.
What sort of precautions would they go through to make sure that they're not actually inoculating the bread when they're preparing it? So just a couple questions in there about that experimental setup.
Yeah. So the first question is about replicates. It's not necessarily critical to have replicates as long as we are doing this as a qualitative activity. And I think the concept of having replicates are become more important if we're trying to measure a variable and we're more concerned about the variation of our measurements. So, for example, if you're trying to measure, for example, the size of mold colonies or the percentage of coverage of molds on the surface of a bread, which are those variables that can be measured and collected as data, then it's kind of important that we have replicates so that we have better graphs on the variation. Because, well, again, we're getting more and more deep into statistics now. The actual response of the experiment, for example, extent of mold growth, can be affected by the factors that we are applying, let's say temperature and moisture. And it can be affected by randomness or luck. So having replicates help us amplify the effect of our factors and reduce the interference of just pure randomness or luck. So yeah, and if students are comparing their samples, it may be a good idea to have them share their moldy bread on social media, like maybe you can have Instagram albums of moldy bread, just an idea, but they can be replicates for each other's experiments, right? So presumably they're using similar conditions and incubating the bread slices for a similar amount of time, then yeah, so that'll still work too. Yeah, that's all I can think of. Probably that addressed part of the questions.
And then, so the second part of the question is, really around handling the bread and
right.
making sure you're not accidentally inoculating a piece, especially if you're cutting it.
Yep. Right. That's a good point, and I probably didn't address that enough in the lesson plan. It's always recommended that you wash your hands with soap and running water prior to the activity. And honestly, there isn't a very big need in sanitizing all surfaces, unless the surface is not particularly soiled. Because, well, that's well, I take that back. That probably doesn't hold true for the control sample with no swabbing. If students need a clean surface to basically break the breads or handle the breads, they can use a piece of saran wrap that are freshly out of the roll. So that basically gives the students a reasonably clean surface to work on. If they have access to any sanitizing reagents like spray bleach or the spray peroxide, that works too, although I don't recommend those because they may interfere strongly with the actual experiment itself, because those disinfectants may stay in the bread. So yeah, saran wrap is usually good enough, or alternatively, if they're pulling a new Ziploc bag from a package, they can just handle the bread on the surface of that Ziploc bag. Yeah, I don't think it's necessary to teach students the rigorous aseptic techniques in this particular activity, because again, it's a little bit complex and the equipment demands is probably also substantial. But yeah, essentially washing hands is probably good.
Excellent, thank you. It's good to know that this sounds like an easy, because you don't have to worry about that. It sounds like this could be something easily done for students at home.
Right.
There was another question up in the chat that I just wanted to bring up because I'm very interested in it. The question revolves around fresh baked bread, or say bread you would get from a bakery, and bread you'd get from a store. I've noticed, as well, the question said that the bread that you get from the bakery is going to mold faster, when left out as opposed to your store-bought bread. I've also seen the way store-bought bread now is usually in 2 plastic bags. There's a loose plastic bag, your normal bread bag, and then there's a tighter plastic bag that's around the bread itself. And that bread seems to last forever. Maybe I just buy highly processed bread, but what is happening there, and why does this occur?
Yeah, so um, it has to do with the packaging method. And so the bakery bread, when they're baked, they tend to sit on a shelf until well, consumer arrives, and then basically the consumer just or the baker just puts the bread in a paper bag or some sort, and then you take it home. So during this whole time, the bread is exposed in the atmosphere, and the spores can just land on the surface of bread. Now, processed, or well, if you buy like bags of sliced bread, so those packaging are done at an industrial facility, and to maximize shelf life, so those facilities usually send the bread directly into a package, and sometimes in an aseptic fashion. So there is much less contact of spores and bread prior to packaging. So that's one factor. The other is the sealed package also helps, because if there's no mold spore landing on the bread, there will be no growth. So in theory, if the bread is freshly baked and then packaged immediately in an aseptic fashion, and then you don't open the bag, it should last forever. There should be no growth of mold at all. So yeah.
Thank you for answering that question. That it's always been something I've wondered, and I've tended to notice that bread seemingly is lasting longer and longer these days. So
yeah. Well, if you read the additives, you probably will notice the use of antifungal reagents as ingredients. Some of them are natural or food grade stuff, so you probably don't think of them as antifungal reagents. One example is ascorbic acid or vitamin C, which lower the pH and also suppress mold growth, but it won't be labeled as an antifungal reagent. Right?
Yeah, I'm not sure I'd eat something that was labeled like antifungal. It seems
yeah,
not appetizing.
Right.
Well, excellent. I just want to make sure there's if there's any other questions, this is the time to ask them. Either you can unmute yourself and ask them, or put them in the chat. Well, thank you very much, Glenn. We're going to make the slideshow that you presented, as well as the lesson plan, available on our website, and along with the recording of this. So if you missed it, or you want to come back and rewatch it, you can. I do want to introduce Jessica Jansen from Oregon Ag in the Classroom. She is doing a number of virtual field trips. These are great ways to engage your students in a fun ag-based field trip, and evidently they are extremely popular. I heard she had over 1,500 people at her last one, so it's a good way to interact with lots of other students. So Jessica, tell us about the next field trip.
Yeah, so our next field trip is coming up here December 7th at TMK Creamery, which is a farmstead creamery right outside of Canby in the Portland metro area. And it's neat. They have a small dairy, and then they make very delicious cheese. If you haven't had a chance to purchase their product, it's delicious. So that's 10 a.m. Pacific time, December 7th. And that's geared towards grades 6 through 12. And we also have a few other virtual field trips in the works. The main one is a Christmas tree farm. We're actually doing a really neat partnership with our partners in Arizona, the Agriculture in the Classroom program in Arizona, to do a citrus tour at the same time. So we'll be in Oregon for Christmas trees, and then see what it's like to grow oranges in Arizona. So we're looking forward to that one as well. That will be also at the beginning of December. So if you'd like more information, I think I put in the chat here to go and visit that. I see free samples. I wish we could figure that out. That would be awesome. It's delicious cheese, but they are becoming more available. So hopefully wherever you are in the state, you can find it. So if anybody has any questions about that, I'd be happy to answer them. If not, be sure to visit the website and sign up for updates on those. Thanks so much.
And we are working on getting, again, in our virtual programming site, a link to the Oregon Ag in the Classroom virtual field trips. There's also a lot of other resources on their website, including field trips that you can watch the recorded. I think it's been, I've been talking to a number of Smile clubs, and a couple of them have been watching the Christmas tree one and thought that that was really great as we are coming up on the holidays. So with that, if there's not any other questions, thank you very much for coming, and we will be having more PDs coming after Thanksgiving. We're going to be getting those up on our website this week, and I will be sending out reminders. And if in the meantime you have any questions, feel free to contact me through our website or directly to my email. So, thank you very much, and we will see you all at the next one.